There is a phase we skipped over called interkinesis, where the two daughter cells kind of realign themselves before continuing into prophase 2. Some species may have interkinesis, others not so much. But what does need to happen is a recopying of the centrioles to form the microtubular spindle apparatus. In prophase 2, the new spindle forms and the nuclear envelope fragments again. At metaphase 2, the sister chromatids line up at the metaphase plate, and again you can see that they are still a mosaic of red and blue from crossing over and from independent assortment in meiosis 1. At anaphase 2, the sister chromatids separate, and in telophase 2 and cytokinesis, we finish with 4 haploid daughter cells, each with 1 copy of the 3 chromosomes. So let's compare mitosis and meiosis. Mitosis doesn't change the number of chromosomes and produces cells that are genetically identical to the mother cell. Meiosis always starts with a diploid cell and always ends with haploid cells. It produces new combinations of chromosomes in the daughter cells. In this slide, we can see a side-by-side comparison of the happenings in mitosis and meiosis. These are cells from the same organism which has 3 homologous pairs of chromosomes. As you can see, 2n equals 6. In mitosis, the homologous chromosomes are present, but all the chromosomes line up at the metaphase plate. So at metaphase, we see 6 sets of sister chromatids. At metaphase 1 of meiosis, the homologous chromosomes line up, so instead of 6 pairs, we have 3 tetrads. Then in meiosis 2, there are 3 sets of sister chromatids lined up. So metaphase looks different in mitosis, meiosis 1, and meiosis 2. 3 unusual events occur in meiosis: synapsis, which is the formation of chiasmata, and the exchange of segments of the chromosomes in prophase 1. Metaphase 1 has homologs lined up as tetrads, not just sister chromatids. And at anaphase 1, it is these homologs that separate, and not the sister chromatids. So where does the genetic variability come from that makes each of us different? Changes in the DNA sequences, which are called mutations. Mutations make different forms of a gene, which are called alleles. Our parents have different alleles of their genes that they pass on to us, and that we pass on to our progeny, and so on. Not all the mutations that may occur are favorable or even survivable. If mutations are favorable for an organism, if they help them to survive and reproduce in a particular habitat, those organisms are more likely going to survive and reproduce, and those new alleles will become more frequent. Sexual reproduction helps by shuffling the alleles into new combinations. While mutation brings about the differences in DNA sequences, we've seen how meiosis can take this variability and really shuffle it up. Meiosis accomplishes this shuffling in 3 ways: crossing over, independent assortment of chromosomes, and random fertilization. Crossing over produces chromosomes with new combinations of the alleles, or different combinations of the different versions of a gene at a particular locus. We call these recombinant chromosomes. Crossing over happens in prophase 1, as we saw, and the swapping occurs between non-sister chromatids of homologous chromosomes. Crossing over enables the different genes we inherit from our parents to be shuffled into chromosomes that neither of our parents can lay sole claim to. This illustration shows how the process occurs within a single pair of homologs. Remember, blue is paternal and red is maternal. In this case, with just 1 set of homologous chromosomes and crossing over in different places along the chromosomes, we can produce 4 genetically distinct daughter cells. These 2 in the middle here having recombinant chromosomes, meaning they are not identical to either of the parental chromosomes. They're not all blue or not all red. Independent assortment of chromosomes I talked about earlier. When the homologs align in metaphase 1, they don't align such that all of one parent's chromosomes are on one side of the metaphase plate and the others are on the other. They align independently of one another, so maternal and paternal homologs are mixed up into which daughter cells they end up in. Here's another simplified case where we have a cell with 2n equals 4, or 2 pairs of homologous chromosomes. There are only 2 possibilities. Either both of dad's chromosomes are on one side and both of mom's across, or there's a swap. Both of these arrangements are equally likely. Let's pretend for a moment that crossing over isn't happening just for simplicity's sake. When we get to metaphase 2, you can see that we are left with different alternatives for how the sister chromatids will be separated. This means that in gamete formation, there are 4 different possible combinations of how the 2 sets of homologs will assort. At each homolog, the direction the maternal chromosome will go, left or right, is essentially a coin flip, 50-50. So the number of possible combinations of chromosomal assortment are 2 to the n power, where n is the haploid number, or as many possible outcomes as flipping a coin n times. In humans, with a haploid number of 23, the number of possible combinations of chromosomes sorting moms and dads is 2 to the 23rd power over 8 million different ways. Again, that's without considering crossing over. Random fertilization increases the odds of having 2 identical outcomes even more, because any given sperm produced from 8.2 million different combinations fuses with a particular egg produced from 8.2 million different combinations. Again, this doesn't even include crossing over, and already the likelihood of having 2 of the exact same outcomes is 1 in 70 trillion. So to put that in perspective, the Earth's current population is 7.8 billion, or 7.8 times 10 to the 9th power. 70 trillion, which is the number of combinations of the fusion of sperm and egg, is 7 times 10 to the 13th power. Thus, there's enough variability in our uncrossed over chromosomes combining into the zygote to form you or me or whomever to make sure that even if the Earth's population increased 10,000 times, there would be nobody on the planet exactly like you. So, to bring it back full circle, when Mr. Rogers said that you are special, you are unique, and there is no one quite like you out there, he wasn't just trying to burnish your self-esteem, he was stating a biological fact. Our learning objectives once more. And the second part. And in our next chapter, we'll meet an Austrian monk with a penchant for gardening and note-taking named Gregor Mendel.